All right, here is another picture on the left of our paramecium, and you can see that this depression here is what's called the oral groove. The oral groove, like we said, is lined with cilia so that when there's a current, food moves into the oral groove, but when there's not a current, those cilia can move back and forth and they can pull it in. The mouth pore is the opening, the groove is the depression. Now, the mouth pore opens into this area here, which is called a gullet. A gullet is just sort of like a holding place. It's sort of like your stomach, only your stomach has digestive enzymes in it, and the gullet really doesn't. What happens at the end of the gullet is the gullet forms these little vacuoles, food vacuoles, and the food moves into the food vacuole, and that's where the digestion begins. So the food vacuole is forming off of the gullet, and they're capable of actually moving. There's one here, there's probably one here and here. They can move once they pinch off of that gullet, they can move throughout the body of the paramecium. There are enzymes in those vacuoles that digest the food into a lot smaller molecules, and the smaller molecules can actually enter into the cytoplasm. Okay, here's another picture. This one shows all the cilia. So the oral groove is the depression. Here we have our cilia. And you also have, this is called a contractile vacuole. We'll talk about it in just a minute. But the undigested materials will actually move from the cytoplasm into the anal pore, which is here. Now, the anal pore has an opening to the outside, and that's the way they expel their solid waste. It's a big deal for an animal to have two openings, one for intake of food and one for the elimination of waste. If we get to the animal kingdom, and I hope we do, we'll find that there's some animals like jellyfish that just have one opening, and so their food intake and their waste expulsion is out of the same opening, which is the mouth. Well, for a little single-celled organism to have an opening to expel the solid waste is a big deal. Now, the liquid waste, here he is in movement. Very ominous music for a little paramecium. Okay, now one of the things that they have are called contractile vacuoles, and the contractile vacuole is used to remove excess water. In paramecium, they actually can collect, because they're in water, so they have an excess of water, and they collect the excess in what's called a contractile vacuole. And when too much water comes in, they can squeeze that contractile vacuole. It's contractile, and they can get rid of their water. For us, we get our water through osmosis, and so as long as there is a higher concentration and lower concentration, we're going to be able to regulate our water intake. But with ciliates that are in fresh water all the time, they only can control their water intake through what's called a contractile vacuole. Now, this is showing you what is called a trichocyst. A trichocyst is an elongated, very cylindrical body that is used for defense. So here we have, this would be the inside, and here we have the outside. Now, I'm not going to ask you the parts of a trichocyst, but they're embedded in the pellicle. Again, the pellicle is that tough layer on the outside of the cell membrane, and there is a very a long spike that comes out of the body of the trichocyst, and it comes through the pellicle. It's spine-like, and they're not really sure what their function is, but as much as it's been studied, they have surmised that it's defense. All right, ciliates also have two kinds of nuclei, and the two kinds of nuclei are going to be a very large nuclei called a macronucleus and a much smaller one called a micronucleus. The macronucleus contains a lot of copies of the DNA that's directing its metabolism and development. So the large nucleus really has nothing to do with reproduction. The small micronucleus that you see there participates in the exchange of the genetic material during conjugation. Now, before in bacteria, we talked about conjugation as a way of actually just transferring that plasmid to make the bacteria more genetically variable. But that's not the way it is in a paramecium. In a paramecium, the conjugation is actually a form of reproduction, and it's the micro or the small nucleus that contains the DNA that actually gets exchanged during that process.